I'm Stephen Hill from the MLC Biostatistics Unit at the University of Cambridge in the UK. I'm going to talk to you about a semi-supervised approach for learning the structure of causal molecular networks. And this is joint work with Chris Oates, Duncan Blythe and Sash Mukherjee. We're often interested in molecular networks where edges in the network represent causal relationships between molecular components, for example in protein signaling networks or gene regulatory networks. These causal networks are in contrast with, for example, gene co-expression networks, where the focus is on associations or correlations rather than causation. So here we're interested in learning the structure of causal networks from data, and doing this in specific disease contexts can help our understanding of disease mechanisms and inform therapeutic targets. I'll just say a bit more about what is meant by a causal network. So these are networks with directed edges that represent causal relationships, and in particular a causal edge from node A to node B indicates that an intervention applied to node A would lead to a change in the abundance of node B. So it's really capturing changes under intervention. There's a distinction to be made between direct and indirect causal edges, and this is relevant for the results I'll show. So an indirect causal edge is a causal relationship that is mediated via other measured nodes. So here we have an indirect causal edge from nodes A to C, and this is indirect because the causal influence of A on C is mediated via node B. But note that direct causal edges are only direct relative to the set of nodes in the network. So here we have a direct causal edge from A to C. It's direct, so the influence of A on C is not mediated via other nodes in the network, but it could still be mediated via some other unmeasured entity, as shown here. So different methods have been proposed for learning causal networks, and the common approach is to use methods based on causal directed acyclic graphs. These approaches have a formal underlying statistical model with explicit causal assumptions, and these are typically very strong assumptions. Here we take an alternative approach and view causal network learning as a machine learning task. So we want a discriminative classifier that can tell whether a causal edge exists or not between a pair of variables. And the classifier takes all available data that we have as input. Before I go into a bit more detail on the method, I'll first introduce a bit of notation. So we have p variables, p nodes in the network forming vertex set V, and then a set of directed edges forming a set E, and then a directed edge from node I to node J in E means that variable I has a causal influence on variable J, and this can be direct or indirect depending on the context. We then have a matrix A, which is the p by p binary adjacency matrix for the network, and then a vector Y, which is simply the vectorized adjacency matrix, which we index by k. So yk corresponds to a particular entry, ikjk, in the adjacency matrix. And we have a matrix B, which is the available data on our variables, and this is the usual n by p matrix of, for example, gene expression values. We then have phi, which is a priori background knowledge on causal relationships, which can come from, for example, databases or other interventional datasets. So here's an overview of our approach. So we want to construct an estimator for the entries of the adjacency matrix, y hat. Remember, y is the vectorized adjacency matrix. And this is a function of the available data D and the background knowledge phi. So we're treating the entries of the adjacency matrix as labels in a machine learning sense, and the background knowledge phi is used to derive a set of a priori known labels. So some entries of our adjacency matrix are known, and the task is to learn the unknown labels. So this fits naturally into a supervised learning or semi-supervised learning framework. And the potential advantage of a semi-supervised learning framework is that it makes use of the unlabeled data in model training. And you can see in this toy example at the bottom with two labelled points, one from each class, that taking the unlabelled black data points into account is very informative for determining the decision boundary, which should be circular instead of the linear separator, which would be a natural choice based on the labelled data alone. So how do we frame causal network learning as a semi-supervised learning task? Well, the entities to be classified are variable pairs. So for each variable pair, we want to determine if there is a causal edge or not. So we need a set of features for each variable pair. We denote the featurization for variable pair k by a function gk, which is a function of the n by p data matrix. We let y superscript l denote the vector of known labels derived from the background knowledge phi, and yu be the vector of unknown labels to be learned. So we need a semi-supervised estimator for the unknown labels yu, which takes as input the featurization for each variable pair and the known labels. So we could essentially use any combination of featurization G and semi-supervised learner. So here we use a bivariate featurization and graph-based manifold learning. For the bivariate featurization, we construct features for each variable pair using the n by p data matrix D and bivariate histograms. So if we consider a single variable pair with index k corresponding to entry ikjk in the adjacency matrix, then essentially what the featurization does is it plots the data for the two variables against each other. So we have variable ik on the x-axis, variable jk on the y-axis, and then we form a bivariate histogram from the scatter plot. The feature vector for the variable pair is then the vector of histogram counts, so this matrix vectorized, and we denote this by xk. So we do this for each variable pair, so we have p squared vectors of histogram counts, one for each variable pair. So the idea is that the featurization captures patterns in the scatter plot that are indicative of causal relationships, and these patterns can be learned by the classifier. For the semi-supervised classifier, we follow the manifold regularization approach by Balkin et al. And here I'll just give a very brief overview of this approach. So we form a p squared by p squared similarity matrix W for the variable pairs, given by this similarity function here. So the xk and xk prime are the feature vectors for two variable pairs, and essentially this is quantifying the similarity between the scatter plot for one variable pair k and another variable pair k prime. We then calculate the graph Laplacian matrix L, which has this form here, where D is a diagonal matrix, where the diagonal entries are the row sums of W. So here we're assuming that the data lies on a lower dimensional manifold structure, which is encoded via this graph Laplacian. We want to estimate a classification function f, which takes a feature vector for a variable pair as input and returns a real value, where the sign of this value agrees with the label for the variable pair, where this is known from the background knowledge. And the function also takes the manifold structure encoded in the Laplacian into account. So if we let f be the vector arising from evaluating the classification function for each variable pair, then the Laplacian regularized least squares approach, as this form shown here, it's optimizing for a classification function f that minimizes the distance between the values predicted by the classification function, so the vector f here, and the known labels y. but subject to some regularization constraints. So the second regularization term here is an ambient regularizer that ensures the problem is well posed. You can think of this as like the regularization term in ridge regression. And the first regularization term is where the Laplacian matrix comes in, and this ensures the smoothness of the predicted values given by the function f with respect to distances between variable pairs on the manifold. So this is where the unlabeled data is used. And this objective function has a closed form solution. So on to some results. So this is data from Cameron et al., which consists of a large number of gene deletion experiments with corresponding gene expression measurements in yeast. So we consider 50 genes as network nodes and interventional data, where each of these 50 genes was knocked out in turn. And this interventional data was used to define a gold standard reference causal network. And this is our background information phi. So for example, if we take the first of the 50 genes and knock it out, and then you can see which of the genes have a change of value under that intervention. For example, if the second gene has a change in abundance, then we can say there is a causal edge from the first gene to the second gene in the reference network. So the focus here is on indirect causal effects, as the intervention experiments can't distinguish between direct and indirect effects. Then our dataset B comprises 80 observational data samples and a varying number of interventional samples, where importantly these interventions do not target any of the 50 network nodes. We then use a fraction rho of the reference network as known labels, and we apply the semi-supervised approach to learn the remaining labels. And our predictions for these unknown labels are assessed against the held out values in the reference causal network. We compared our approach, which is called MLCL for manifold regularized causal learning, with correlations, lasso regression, k-nearest neighbours classifier, and IDA, which is a causal directed acyclic graph based approach. So here are the results, where we used area under the ROC curve as the performance metric. So each of these three facets is a different training data size, shown at the top here, increasing from left to right. And the x-axis is rho, the fraction of the reference network that was used as known labels, going from 0.2 up to 0.8. So the MLCL approach, shown in red, performed well relative to the other methods, with improving performance as rho increases, as we would expect, as this increases the proportion of the reference network which is known. And it even performs well when only 20% of the reference network is given as labels. K-nearest neighbours, shown in yellow, performs a bit better than the others, likely due to the fact it also uses the known labels, whereas the other approaches do not. Here's another example using TCGA protein data. The setup is similar to the last example, with the main differences being that this is observational data only, there is no interventional data, and the reference causal network is derived from literature, so it encodes direct causal relationships. So here we're trying to learn direct causal effects instead of indirect effects, as in the last example. So the results are broadly similar to before. We can see that MLCL is again performing well, even though this is now just observational data. And another difference is that k-nearest neighbours is no longer performing a bit better than the other methods. So I'll finish with some concluding remarks. So our approach showed good empirical performance, in our experiments relative to some other approaches that might be used for the causal network learning task. The output of our approach doesn't need to be acyclic, and the edges may encode direct or indirect causal relationships depending on the precise input, as we saw in our examples. The approach doesn't require an explicit causal model, so patterns in the data that indicate a causal edge are allowed to emerge via the learning procedure. Our output is, however, less rich than a full causal model, so we're really just trying to do binary detection of causal influences. Do they exist or not? We're not able to estimate, for example, the magnitude of causal effects. You can find more details of our work in this manuscript, including some theoretical results by Chris Oates, who's a joint first author on this work, and further empirical results also, and code for the method and for the empirical investigations can be found on GitHub. So I'll finish by thanking my collaborators on this work, Chris Oates from Newcastle University and Duncan Blythe and Sash Mukherjee from the German Centre for Neurodegenerative Diseases. And thank you for your attention.